let's go ahead and get started. So we're going to be doing not one, but two topics today. All right, my usual plug to that thing, Think Fiveable, Instagram, Twitter, all those. I am not on Instagram. I am on Twitter. I post a lot of science stuff on Twitter. My students like to tag me in science memes, and it makes me really happy. So if you ever see a good science meme, feel free to just click, link, because they give me joy. There are so few things in life that give us joy, and I have found mine. It's science memes, so go figure. But also follow that one up there, Think Fiveable. Sorry, I had to cover my face. Think Fiveable. We love to see what you guys are doing, what you're liking, what you're not liking, anything that can help us grow, anything you think we should keep doing. And so if you're ever hanging out, taking your notes like a good student, here's my notes because I'm such a good student. Share them with us. I know some people are really into doodle notes. Here's what you actually came from, science. So we just did our cute little intro where I plugged all the things about me and Fiveable. Gonna talk about compartmentalization, compartments. It's like the container store, your cells. They all have their individual little containers. If you've never been to the container store, find one. It's awesome. But then we're also going to talk about the origins of cell compartmentalization. So when did it start doing that? Why did it start doing that? And full warning, when I was in college studying biology, I spent a lot of time on this. So I'm going to nerd out, and I'm going to need you to nerd with me. So thanks. And then, like I said, the part most of you want, some AP practice, some real old questions. Some of them are pretty old. I think I had to go back all the way to 2002 for these. So sorry, the format may be a little different, but it's the same information. Sorry, I have had a long day, and so now I'm checking out my hair in the webcam, but now I'm done because I'm a mature adult and I know I need to do my job. Here we go. Way too much information, y'all. I'm sorry. I've had too much sugar to try to stay awake. So I'm going to make this a little bigger so y'all can see. I have two scenes here because I like to start things off with a metaphor. Why are we compartmentalizing? So what I like to think of whenever I think of this, I think of two different things. I think of the Parent Trap. Have you guys seen the masterpiece that is The Parent Trap? The Lindsay Lohan version. I mean, the other one's fine, but the Lindsay Lohan version is better. Have y'all seen The Parent Trap? Yes? No? Maybe so? It's okay if you haven't. Of course! Thank you, Anna. Okay, so in The Parent Trap, when it starts out, these girls are at summer camp, and the girl pops out from behind the luggage. She's like, I'm here, I'm ready, summer camp, woohoo, I have a twin. But there's this pile of luggage. And if you remember, they have to be like, yo, tie-dye girl. And that's how they get their stuff out from there because it's so disorganized and it's just a heap of stuff. And it's not organized. It's hard to find things. But if you've ever ridden an airplane or a bus or anything like it, airplanes always have these bins overhead. The bins are where you store your luggage. It's meant to make things very, very organized. It's meant to. Sometimes you get people on airplanes who do not play the organization game, and they're not fun. But for the most part, it's pretty good. Because, when you get on the airplane, everyone puts their stuff over their chair. And then when you get off the airplane, you get your stuff down and you file out orderly. And it's a very orderly process. It's not necessarily the fastest, and that's where the metaphor ends. But everything is organized. Everyone has their own little compartment for their stuff. We like cells to have their own little compartments for their stuff. It makes us happy because they're organized. Like I said, if any of you have ever been to the container store or the organization section at Walmart, that's like my place. But cells like things to be organized when they can because it makes them more efficient. So I'm going to make my face bigger so I can emphasize this to you. In this unit, structure and function, you're going to talk about how structure impacts function. You're also going to talk about what makes cells work efficiently or what makes them function efficiently. So in the past, you've talked about cell size. So remind me, I'm going to quiz y'all on the fly. Which type of cell is more efficient? A big cell or a little cell? Baby cell. Which one's better? Big cell. Small cells are more efficient. Very good, Mark. So it's going to be about efficiency here. Why do we have compartmentalization? It's because it's more efficient. It's like why your mom always makes you clean your room. She thinks it's going to be more efficient. If you're like me, you have organized chaos and you only know where things are when it's dirty. But wait till you're a grown-up. That's actually a problem. We like compartmentalization because it makes it organized and it makes it easier for things to do their job. That's why we like small cells. It's more efficient to move stuff in and out, in and out, and that's more efficient. So the theme here, as you can see, is efficiency. We want things to be efficient. So blast from the past, the way that we're gonna look at organelles now. So here, I'll make this a little bigger. Sorry guys, I just like looking at my own face. So, because I'm always scared I'm making like some crazy eyes. And so I'm like, okay, I gotta watch myself in case I'm doing something nuts. But going back to organelles, if you remember, there's a certain type of cell that always has membrane-bound organelles. What type of cell is gonna have membrane-bound organelles? Eukaryotes. Very good, Shreya. Hi, Shreya. I'm so glad you're here. Eukaryotes, the more modern cells, and we're actually going to talk about how they form today, but they're going to have a membrane-bound organelle, and that means they literally have phospholipids all around them in a cute little phospholipid bilayer hug holding their stuff together. And so same as with the cell membrane, stuff's going to cross that the same way with the passive transport and da-da-da. I'm not going to get into that because I already taught 2 lessons on that. If you want to watch it, you can click that little button, those 3 little bars, and go to past streams and be a premium member and watch them there, but I'm not doing it again. But in any case, it's got that membrane and it's gonna have to go through the membrane in order to get into those organelles. So we've got a Golgi, lysosome, and endoplasmic reticulum. These are gonna be your single membrane organelles. They have a membrane, but it's just one layer. Uno. I don't know how to say one in any other language, just one and uno. Sorry, I need to get on Duolingo, but I don't because I'm busy doing stuff like this instead or watching Netflix. So, sorry. But then on the other hand, you have these 3 little nuggets, a mitochondria, a chloroplast, and a nucleus, and they have a double membrane, 2, dos. I don't know another word for 2 in another language either, sorry. But they have a double membrane. And so there's a couple reasons for that. When I think of the nucleus, I think of it as being extra security. So you want that double membrane to have that extra security because you're protecting the DNA. But when it comes to the other guys, that is the mitochondria and the chloroplast, that's going to get really important in a minute because that's going to increase our surface area. And remember, do you want to have more surface area or less surface area? More. Very good, Tria. We want to have more surface area. Very good, Soleil. And I apologize if I mispronounce any other names. There's no phonetics next to them. So if at any point I am butchering something, please let me know. I don't want to offend anyone. But so these double membrane organelles, they are going to have that increased surface area. If you saw one of my other streams on this, because I've been on here a lot lately, I don't think I have it in this notebook because this is actually mine for my school. But I drew out the cell and I got like my measuring tape and I measured it for you, like how much increased surface area it is when it has that inner layer. That's a lot and it lets it do a lot of stuff. So another pop quiz, what do we call that inner membrane of the double membrane of the mitochondria? Where it's doing all the cellular respiration stuffs. Starts with the, C. Starts with a C. Cristae, very good, Shreya. Okay, so that inner layer in here is going to be called cristae, and that's going to be your increased surface area. So we're going to talk a little bit more about that as we go on. But just a reminder, those eukaryotes do have membranes around them, and that double membrane that we see over here is going to get real important in just a minute. So the other thing about compartmentalization is we like compartmentalization because we can compartmentalize, sorry, that word's hard to say, processes. So I want you to think of it this way. You have a house. I assume you live in a house. If you don't live in a house, sorry. But I assume you live somewhere that has a place to do dishes, place to brush your teeth, place to sleep. There's probably some other stuff in there. But you have a house and you do different functions in the parts of your house. You have a kitchen where you cook. You have a bathroom where you bathe and do other stuff. You have a bedroom where you hopefully sleep. Don't fall asleep on the couch watching Netflix too much. It's bad for you. But you have different places where you do different things, and it's efficient that way. If you live in, say, a studio apartment, which wait till college, you're gonna love it. But then you have to do all your processes in one space. A studio apartment is basically one room where they squeeze in a kitchen and your bedroom and your living space. And luckily, they will put the bathroom like behind a wall, but it's technically still considered one room. And it's kind of backwards. It's called an efficiency apartment. That's how it'll be advertised, but it's really not that efficient. You've got everything everywhere. No one knows where your kitchen ends and where it starts. And it gets real messy real quick because it's small. And if you're like me, you have a lot of stuff. So we prefer when we have a house with walls where we can put all our different processes in different places. Cells are the same way. So if you look here, you've got a bunch of different processes going on, but I want to do a quick reminder on enzymes and proteins, because if you're like me, I haven't actually taught enzymes and proteins in my brick and mortar class yet. That's our next unit when we get into energetic. So I want to go ahead and do this refresher in case any of my kids watch this later and they're like, see, what were you talking about? But so you've got an enzyme, and remember, the only way for an enzyme to work, it has to fit with a substrate, and they have to match like a lock and a key. If something doesn't fit, if it's the wrong shape, it's not going to work. But that's going to make an enzymatic process or a catalyst, and it's going to spur some metabolic something that is hopefully good for you. So that's the quick and easy version of it. But there's a bunch of different processes in your cells that are going to be really important. You've probably already kind of touched on photosynthesis, this green one that's happening in the chloroplast. You keep photosynthesis in the chloroplast. We want it compartmentalized there. We want it separated. We want it nice and pretty in its own little room with no one to bother it. It's like when you're practicing your oboe. You don't practice your oboe in the middle of the kitchen when everyone's trying to eat dinner. You go into another little room and you practice by yourself where you can focus. Same thing with photosynthesis. You stay in the chloroplast. Over here, we've got mitochondria, and we've got a cellular respiration process going on. We've got the glucose entering the cell, and then it's all going into the mitochondria, more into the details of that later, specifically next unit. But it's a compartmentalized process. It's happening in the mitochondria. It's not like happening in the bathroom while someone's taking a shower, because that would be awkward. It's got its own separate room and its own separate place, and that's how it does it. Compartmentalization. Final example, if you're like me, you won't get to this till like the end of the year. It's called protein synthesis. Your teacher may call it the central dogma. You will soon call it the central I hate it. But then you'll be like, OMG, I love it because everything makes sense now. Just wait, it's magical. I'm nerding out a little bit right now. But talking about protein synthesis, so you can see right here, we've got some DNA. Where does DNA live? In the nucleus. Very good, Tria. So that enzymatic process is starting in the nucleus. And then you can see it leaves the nucleus, and then it's going to go out here. And we know proteins and things are packaged in Golgi bodies and endoplasmic reticulum, so it's going to move out there. But either way, we're sticking to very specific organelles to do very specific jobs. One other thing I want you to notice, mitochondria and chloroplasts as well. Yes. Wait, Soleil, let me confirm. What do you mean mitochondria and chloroplasts as well? Like just they're compartmentalized in those two organelles? Having DNA? Yeah, we're going to get there. Okay, you're thinking a little ahead. You're putting a wrench in my plans. So hold that thought for just like a sec and stick it back in your brain behind your ear. But very good foreshadowing. I appreciate that. We're going to get to that. But what I was actually going to say is we start with DNA in the nucleus. We start in the chloroplast. We start in the mitochondria. What do all of those have in common besides DNA? I just showed you on the last slide. What do they all have in common? What are the double membranes? Very good, Shreya. All three of these, nucleus, chloroplast, mitochondria, they all have double membranes. Huh. Interesting. We're going to learn more about that in a minute. So you can see here, this one's unlabeled. Sorry, I did the best I could. But we got our mitochondria, we've got our chloroplast. So again, we've got this kind of dark orange membrane. And that's our outer membrane. And that's kind of like the shield that's hiding it from the rest of the stuff. But then in here on the inside, we've got that crease dye, and that's the increased surface area. If I was to take out my magic measuring tape and measure this, which I'm not going to do because all your computer screens are different, and so we wouldn't all get the same results, and then the data would be invalid. But then it would show there was increased surface area compared to if we just went around the inside of the outer membrane. We want that increased surface area so we can do more cellular respiration, so we can make more ATP, because time is ATP if you're a cell. It's not like S for time is money. However, as Soleil pointed out a minute ago, notice this little squiggly line right here. Itty baby squiggle. What's that little squiggle look like to you? If you were a scientist, what would you diagnose that little pink squiggle as? DNA. Very good, Sandra. In fact, if you're like me, you just looked over here and saw, there's a blue squiggle and it's called DNA. These both have DNA. Do those DNA have nuclei around them for the mitochondria and the chloroplast? No. Very good, Shreya. Spoiler alert, there's no nucleus. And what does it mean when a cell or an organelle has DNA with no nucleus? What's that mean when you got this DNA with no nucleus? It's just a tangled up little circle. It's a prokaryote. Very good, Shreya. OMG! I'm sorry, I know a bunch of you have already learned this, but it still hypes me up every time. They're prokaryotes, except actually they're inside all my cells and I'm a eukaryote, so what's going on? What? I hope you're all as hyped as me from wherever you're sitting or laying. Interesting. And so going back, just a little review on the surface area. Sorry, my slides are a little out of order here. We do have that squiggly DNA in there somewhere, but remember, we want that increased surface area because when we break one big cell up into a bunch of little bitty cells, it's going to give us more surface area, specifically this cristae in the mitochondria. So this is going to take way more time to go all the way around and way more space to do cellular respiration than if I just use this outer circle. Same thing if I'm going through the chloroplast. I don't feel like I talk about that as much, I'm sorry. But chloroplasts, they're a little different. They don't have like that just squiggly lines of cristae. They've actually got stacks of things called grana, which are made of thylakoids. But it's the same concept. They do have that increased surface area so they can do more photosynthesis. Justin, endosymbiosis. Ooh, someone's thinking ahead. All right, we're going to talk about that. Put that little thought right behind your ear. Hold on to it for just a sec because that's in the next couple of slides. So membrane-bound organelles evolve from once free living prokaryotic cells via endosymbiosis. So I want you to put on your little, any Harry Potter fans in here, put on your little time turner, and I want you to flick it back a bunch to like 4 billion years ago. And 4 billion years ago, so we're turning back time, you would arrive and you would see these big rocks. They wouldn't be as big then, but they'd still be there because 4.5 billion years ago, I don't know if you've studied what Earth was like. It was not pleasant. I don't know if you've ever seen the Star Wars movies. We're on that like fire planet where Anakin gets all burnt up. Spoiler alert, in case you haven't seen Star Wars. Sorry, don't watch anymore because it's only gonna get worse. But on the fiery planet, it's how you would describe as the 7 layers of the bad place. It's just fire and sulfur, and I don't really know how they breathe there. I think George Lucas took some artistic liberties there. But early Earth was the same way. Like when we first started this cute little planet, there was nothing alive on it. It was just hot balls of rocks from lava, and there was sulfur and carbon dioxide in the air. There was no atmosphere as we know it. There was no oxygen in it. So it wasn't a great place to be, but after about 1 billion years, so now stop going backwards, go forward. So return your time turner 1 billion years forward, and life started. We're not going to get into the origins of life right now. That's more for like later. But what I am going to talk about is after it did form, it started with prokaryotes. You know, we had those. They were the simpler ones. And one of the earliest forms was something called a cyanobacteria. We call them cyanobacteria because they were like a cyany bluish color. You can actually still get them. I studied them in college. We're trying to put them on the moon right now. It's not the moon, Mars. It's fascinating because cyanobacteria were some of the very first organisms to do photosynthesis. And when you stack them up, they actually form these rock thingies. So they look like rocks coming out of the ocean. And if you look at them, they always kind of curve underneath. And I'm sorry, I always forget what the rocks are called. I think they're called stratolites. But if you cut them open, they actually create these really pretty ridgy patterns. And there's fossils of old, 3 to 4 billion year old cyanobacteria inside. So it's fascinating. I told you this is my nerd out part. I'm sorry, guys. It's amazing that you can see and fossilize 3.5 billion year old bacteria, because that was the first bacteria we had. And this stuff was amazing because, like I said, it was the first photosynthetic organisms. So what did it have? Chloroplasts. Very good, Shreya. They had chloroplasts, and those chloroplasts, it's amazing. They were able to take all that carbon dioxide out of the air and turn it into oxygen. These guys are the guys that are actually responsible for giving us the oxygenetic atmosphere that we enjoy today. If we didn't have that, life as we know it would not exist. It's incredible. The bacteria created the blue sky for us. They created that oxygen. And so I'm like, I owe you so much, bacteria. Thank you so much. So yeah, but anyway, coming back to the endosymbiosis and coming back to the chloroplasts. So these cyanobacteria were basically just, they were cells, they were prokaryotes, but they were basically just the chloroplast, and that was it. They had a membrane, they had the thylakoid and the granite and stuff like that. But, so if you look here, sorry. So it was just a very simple prokaryotic cell. It had little squiggly DNA, had some plasmids and some of the materials that would make a chloroplast, but they just had a single membrane. So I bring up Lynn Margulis because I always try to highlight famous scientists, especially when we run across female scientists, because I feel like we should always acknowledge them. So Lynn Margulis is the scientist who actually is known for the theory of endosymbiosis. It's huge, and the more you study it, the more you'll see it and everything, but she's the one who came up with it. So I always try to put her picture here and be like, hi Lynn, how are you? But so what she found was we had these old prokaryotes, and they started to form these nuclei. They started to get like these extra membranes for whatever reason, whatever mutation. We know that these mutations occur randomly. Sometimes they're good, sometimes they're bad, sometimes they're just whatever. But there were other prokaryotes, and they were cyanobacteria and aerobic bacteria. So they had the mitochondria. And the theory goes basically that they did some endocytosis. So basically they went chomp and swallowed the mitochondria and chloroplast. But as we saw, mitochondria and chloroplast have their own DNA. So the mitochondria and the chloroplast kept dividing, and the prokaryotic cell kept dividing. And over time, they formed this symbiotic relationship where they relied on one another. The mitochondria and the chloroplast would supply energy, and that big cell would provide protection. And it's this beautiful relationship that then, so keep in mind, once these cells, cyanobacteria and aerobic bacteria, entered the prokaryotes, that then formed this double membrane around them. That's where their double membrane came from, because they were absorbed. And you know, when you have endocytosis, you've got this vesicle that'll form around it, and it made a double membrane. Ah, science. And that was also around the time when cells started getting a lot more efficient. Don't get me wrong, they could do they could have mitochondria, they could have chloroplasts, they could do cellular respiration, they could do photosynthesis, but they weren't as efficient because they didn't have as much surface area, because they only had one membrane. But now, once they were absorbed, they had two membranes, extra surface area, extra room to do stuff, activities, if you will. And so, dun, 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 that's when we had a big bang, basically. Life just started exploding, because all of a sudden mitochondria had all this increased surface area, so it could make more and more and more and more ATP. It started being able to do more stuff, and that was critical, because the more energy you have, the more it can divide. That means the more it can mutate, the more it can change and adapt and evolve, and it gets real cool. Sorry, I'm nerding out. So basically, like I said, you had this prokaryote, it got eaten up by the eukaryote, or eaten up by a bigger prokaryote, I guess you could say. And that's what made it a eukaryote. And so once that happened, we then had pro and eukaryotic cells. There wasn't just one type of cell anymore. And they did change as they went on. You know, there were some that didn't keep the chloroplast. Like we have animal cells. We don't use chloroplasts. We found other ways to get our energy, and that's by eating the plants, but that's another story. But they get these symbiotic relationships where they work together and they can do so much more stuff. And I think that's a testimony to how we are so much better when we work together. And I know I'm getting a little like kumbaya here, but for real, y'all, just collaborate. You can do 5,000 more things if you work with others. And you can see that in endosymbiosis because you had these teeny tiny little bacteria that got absorbed by some slightly bigger bacteria, and now they can do way more stuff. So whenever I talk about this, though, of course, there's always the kid who will be like, well, how do we know that even happened? How do we know it didn't just like magic? And I don't know. There was probably a witch who did it. And I'm always like, child, if you bring up witches and the flat earth one more time. But I do have to give evidence because I'm a scientist and we rely on evidence. So a little bit of evidence. Some people have already brought this up. Thank you, helpful people. One thing we know, they have their own DNA. And so it's actually pretty cool because if you watch crime shows, one of the ways that they will actually track who your family is they will use your mitochondrial DNA. Mitochondrial DNA is cool though because it's only passed on from your mother. It's on an X chromosome or something, I guess, but you only get mitochondrial DNA from the mother. So it's used to track like parentage and things like that. But it has its own DNA. It divides on its own. DNA, like in your own nucleus, doesn't make the proteins to make mitochondria. Mitochondria does its own business. It makes its own mitochondria in its own little cells. It's all on its own. It's a strong, independent woman, or man, if you will. Chloroplasts are the same way. In plants, they have their own DNA. They do their own DNA replication, their own meiosis, mitosis, all that stuff. I mean, they're prokaryotic, so I guess they don't do mitosis. I guess they do more like binary fission. But either way, they don't rely on our nucleus to do it. They're like the opposite of a virus. They are completely independent. So that's one piece of evidence. You can take them out and they will keep doing their thing. The next one is they also have their own individual ribosomes. And those ribosomes, you guessed it, have their own individual mitochondrial and chloroplastic DNA. So they're highly individualized. Again, that mitochondrial DNA is only shared from the mother, which is fascinating and very handy in science, especially if you want to track parentage. I don't know anything about plant gender, so I can't tell you if that's only passed through plant mothers or plant fathers, but they have their own DNA and they do divide on their own. So we're about to move into the practice AP questions. Perfect timing. We've got about 30 minutes left. So quick recap, they've got their own DNA. They do their own replication. They've got their own ribosomes. We have evidence that they were prokaryotic because they do have their own DNA and because they have that double membrane. Those are going to be the main pieces of evidence. If they had single membranes, then they probably would have developed inside the cell on their own. But since they have the double, that's evidence that they were absorbed via endocytosis. And Lynn Margulis did all this. She's awesome. Good job, Lynn. All right, let's do some practice AP questions. Go team. Dun, 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 dun. Mitochondria are found in most eukaryotic cells and contain their own DNA and ribosomes that are similar to those typical of many prokaryotic cells. Which of the following statements is justified by these observations? A, B, C, or D? Tell me what you think. B, B. Thank you, Shreya and Anna. So an ancestral cell most likely engulfed an aerobic prokaryote in a relationship that proved beneficial for both cells. What do we call a relationship when it's beneficial for both? It's beneficial for both. Mutual. Symbiotic. Okay, so guys, I'm going to warn you just because I have very specific experience with this in AP reading. I graded the AP exam last summer and there was a question on mutualism. I'm going to warn you, if they ask about it being beneficial for both, they want to hear mutualism. They don't want to hear mutual. They don't want to hear symbiotic. There were so many kids I couldn't give points because they said mutual or symbiotic. They want to hear very specifically mutualism. Because if you say mutual, it could mean it's mutually good or mutually bad. So they want to hear that specific vocab word, mutualism. So free information, if you encounter that on a test, that is something that they are very, very, very particular about. Didn't mean to call y'all out, just wanted you to know because I want y'all to do great. But yes, the answer is B. They were engulfed. It is a mutualistic relationship. One other thing I want to point out, it says their DNA and ribosomes are similar to those typical of many prokaryotic cells. I forgot to mention this. So when I say typical of prokaryotes, go ahead and show the answer here. That means it's going to have that circular DNA. There's no nucleus in the mitochondria or the chloroplast, and the ribosomes are prokaryotic. What do we know about prokaryotic ribosomes? What's special about them, or what's different about them? What's different about prokaryotic ribosomes? They don't come from a nucleus. They're bound. They're not necessarily bound. The main thing about prokaryotic ones is they're smaller. The ribosomes in prokaryotes are much smaller than the ribosomes in eukaryotes. Okay. Membrane-bound organelles have an important component in the evolution of complex multicellular organisms. Which of the following best summarizes an advantage of eukaryotic cells having internal membranes? So what's the advantage of that compartmentalization? A, B, C, or D? C. Very good, Shreya. Very good, Soleil. So it is going to be C. I do want to talk here about what's wrong with some of the other ones. It says here eukaryotic cells are able to reproduce faster because of the organelles. That is actually false. Eukaryotic cells take longer because they have to copy all of those organelles, whereas prokaryotes are like, ha ha ha, it's just me. Zoop. So it's not necessarily faster to be a eukaryote. In B, it also says that they are similar to prokaryotic cells in structure. While that is true, it's not necessarily an advantage. It's just their structure. And then D, compartmentalization leads to a higher mutation rate in DNA. Not really. I mean, DNA is just going to do what it wants to do. It doesn't care if it's locked up somewhere or not. So, okay, very good, guys. The following questions refer to the following diagram. Sorry, I know the diagram's kind of small. For each phrase or sentence, select the labeled part to which it is most closely related. Each option may be used once, blah, blah, blah. So which of the labeled parts evolved from a phototrophic prokaryote? B. Very good, Shreya. Very good, Soleil. So it's going to be B. Sorry, I know it's blurry. So B, what is that organelle that's got B? Chloroplast. Very good, Shreya. Very good, Anna. So it's going to be B. It's going to be that chloroplast because that's the trademark of plants, of anything that can do photosynthesis. Very good. Okay, I'm going to go ahead and move on. Last one. Which of the following best supports the statement that mitochondria are descendants of endosymbiotic bacteria-like cells? I see A, I see A. Similar ribosomes and DNA. They're going to have that prokaryotic stuff. A. Do they have nuclei? No. There's no nuclei in a prokaryotic organism. And the microtubules and glycolysis, that's irrelevant to this. So the answer is going to be A. Very, very good. Okay. Oh, I have one more. Haha, surprise. I thought I only had 4. Sorry, guys. Okay, so we got one more. Which of the following is a characteristic of mitochondria and chloroplasts that supports the endosymbiotic theory? So this is kind of like the same question, but different words. Tell me what you think. I see B and I see C. And I see B. And I see B, and I see C. We're all over the place. All right. Moment of truth, y'all. Drum roll it up. It's C. So, and this is from College Board's Key. And I will say, this is one of the old questions I had to dig up from like 2004. So, and that's why it's got a part E on it. My rationale for it being C and not B, because if you think about it, C and B are kind of saying the same thing. Reproducing on their own, having DNA. When cells reproduce, it's because they have DNA to tell them, hey, it's time to reproduce. But that DNA is what causes it. And so that's the root of the problem, so to speak. That's what enables it all. So I would, and keep in mind, they're in symbiosis. The mitochondria and the chloroplast supply the energy for the cell. The cell provides protection for those organelles. I know I said a minute ago, oh, you can pull them out and they'll still do stuff. In a lab, yeah, you can probably play with them and get them to do whatever you want. There's lots of ways you can control that environment, but just in the wild, you're never going to see like a mitochondria sitting around just like, hey, dude, I'm the powerhouse. I got kicked out of my cell. Typically, they're going to stay inside their cell, but they do have their own DNA molecules. So that's why it's going to be C. Does anyone have any questions about that? Sorry, I know that one was a little confusing. I went a little backwards on that. They can reproduce on their own. They do reproduce on their own, but it's going to be inside the cell. That's a very good question, Anna. They reproduce on their own inside the cell. If you take them out of the cell, like in a lab, you can probably make them do whatever you want. But if you like stick it on your desk and you're like, mitochondria, reproduce, it's going to get really shy and it's not going to do it. Does that answer your question, Anna? Okay, very good. All right, y'all. Well, I know what y'all really want from me now. So you, of course, want to see the link to the Twitter and my stuff again, and the super obvious, the link to my slides. I know everyone loves to have the slides, so here's your prize for sticking with me for this 44 minutes. All right, guys, but that's all I've got. So I really appreciate y'all sticking around and participating, and I hope this was helpful. And if it was, please come see me again. I don't know my schedule off the top of my head because I have 5,000 things on my calendar, but you can check the Fiveable website. My name's Jessica. It's on there. So if you want to come see me, hi, I'm here. But I'm going to go to bed now. So you can probably see my eyes are like drooping because I'm so tired. But I hope y'all have a wonderful evening and a great week and a great weekend and a great biology class. So peace out. Bye-bye.